Dear students, in our previous session, in our previous class, we had discussed about single cell protein. That you all know, 1 mark it will be asked, what is the definition of single cell protein? Or for about 2 marks, they'll ask the definition and as well as the applications of single cell protein. Very small topic and a very interesting topic rather. Followed by that topic, today we will be discussing about plant tissue culture. This is the final topic of the chapter. Very important topic rather. Plant tissue culture. You all very well know what are the meanings of this, right? Culture here refers to rearing of plants, breeding of plants, caring of plants is what is known as plant tissue culture. This is a technique above the hybridization technique. So this technique of plant tissue culture acts as a connecting link between plant hybridization and transgenic plants. There are two concepts here. In this chapter, we have discussed hybridization of plants and now we are discussing about the plant tissue culture. Whereas in case of biotechnology chapter, 12th chapter, we will be discussing about transgenic plants. So how to make transgenic plants, how to form transgenic plants, so this technique is the basis for that. So try to understand that because it acts as a connecting link between plant hybridization and transgenic plants. So this technique is what is referred as plant tissue culture. Basically, this concept of plant tissue culture was developed by two scientists. One is known as T.H. Morgan. You might have heard his name in many other chapters also. He is a pioneer in case of mutations. T.H. Morgan, he has observed many evolutionary changes in case of Drosophila also. So this scientist, right, has contribution in many fields. So he is the first person who coined the term, T.H. Morgan coined the term totipotency. Totipotency. Now this word, right, has two words, that is the name of toti refers to total or complete or whole, right? Potency, in case of physics you might have studied potential energy, fine, or kinetic energy. Potency here refers to ability or capability or its energy, right? So here what do you mean by this? Totipotency, total ability, the total concept. So this is a property of plant cells. You don't come across totipotency in case of animal cells. Animal cells are not totipotent cells, whereas animal cells are pluripotent cells. For example, what do you mean by that? In animals, if you take heart cells, if you try to grow on an artificial media in laboratory condition, in vitro conditions, in the laboratory conditions, from heart tissue, heart cells, only heart tissue, cardiac tissue will develop. In case of, if you take the tissue from stomach, only stomach tissue will develop. This is what is known as pluripotency in case of animals. Fine. If you take skin cells, only skin cells are grown. So this is what is the specificity is more in case of animals, complexity is more. Whereas here, in case of plants, there is less specificity, less complexity of tissue. Fine. Here, all the cells, fine, or tissues or organs, they possess a property what is known as totipotency. That means to say that, fine, any part of a cell, any part of a plant, like cells or tissues, when they are grown in artificial media, they give rise to a complete plant of its own kind. Such a property possessed by plant cells is what is known as totipotency. Now I go for the definition. If it is asked in the 1 mark question in your final exam, totipotency, the ability of plant cell or tissues to give rise to a complete plant of its own kind is what is known as totipotency. The term totipotency was first coined by T.H. Morgan in the year 1901. Then one more scientist by name Gottlieb Haberland. In the year 1902, he practically proved the ability of these plants. He thought of this, he coined the term totipotency. He told this concept, he started this concept, but in the lab, in the practical laboratory, it was practically this ability of plants to give rise to a complete plant of its own kind. This was demonstrated by, practically experimented by, shown by a scientist known as Gottlieb Haberland. So the whole concept of plant tissue culture is totally based on totipotency. Hence, this scientist Gottlieb Haberland is known as the father of PTC, father of plant tissue culture. Father of plant tissue culture. From here onwards, from 19th century onwards, the concept of plant tissue culture was started based on the property known as potency. This is the definition you will be asked in 1 mark in your final exam. Clear? So try to understand. So based on the potency, the property of plant cells to give rise to a complete plant of its own kind when grown in nutrient media, artificial media, in aseptic conditions. Now I am coming back to the definition of plant tissue culture. I told you the definition of potency. Make a note of it. Do remember? It will be asked for 1 mark. Now what is the definition of plant tissue culture? The process of growing of plants. The process of growing of plants in a artificial nutrient media. What media? Nutrient media under aseptic conditions. Aseptic, I have told this word in human health and diseases. The condition, the place where it is free from, devoid of, free from microorganisms. Aseptic condition. Under aseptic conditions of temperature and light. This whole process is what is known as plant tissue culture. I repeat, listen once again. The process of growing of plants. The process of growing of plants in artificially nutrient media. Here, nutrient media refers to this media is artificially prepared. It consists of macronutrients like carbohydrates, proteins. It consists of micronutrients like minerals, like vitamins, right? All those things. So such a nutrient media is artificially prepared, taken in a glassware, in bottles, in test tubes. The process of growing of plants in artificially nutrient media under what conditions? Aseptic conditions. What do you mean by that? The conditions that the environment, the place should be free from microorganisms. There should be no bacteria, no fungal spores, or no viral particles. Such a condition under controlled conditions of temperature. Because you have studied in your basic chapters that plants do photosynthesis. They go for respiration. They go for photosynthesis. They need different illuminations of light at different functions. So under aseptic conditions of temperature and light, there is a dark reaction, dark cycle, light reaction. You have studied in basic chapters of first year. So to facilitate all these processes in the plants, we maintain controlled temperature, controlled conditions of temperature and light. Illumination will be set. So after all these, after many weeks together, this will grow into small plants. So the process of growing of plants by using artificial nutrient media under aseptic conditions of temperature and light is called plant tissue culture. Is called plant tissue culture. Now one more definition here. It is very important, right? The terminology here. One is known as explant. Since this is the first class of plant tissue culture, you try to understand the terminology. What do you mean by explant here? As the name suggests, as the word itself is self-explanatory. What do you mean by X? X refers to previous plant, the previous part of the plant that is used in plant tissue culture, the part of the plant that is used in tissue culture. I told you in the previous definition, the potency, cells can be used, tissue can be used, right? Or pollens can be used, or bark can be used, or shoot tip can be used, or root tip can be used, or leaf part can be used. So all these are the examples of explant. So what do you mean by explant? The previous part of the plant. From that particular plant, you have plucked, you have taken some part of the plant, and that you are going to use in your plant tissue culture experiments. Such a part of the plant which you have taken from the plant, original plant, right? And you are using it in your experiment in the process of plant tissue culture. Such a part of the plant is what is known as explant. So that's why the previous part of the plant used in tissue culture. If it has been asked for 1 mark in your final exam for definition, define explant. You can write that the previous part of the plant which has the ability to grow into complete plant and used in plant tissue culture is what is known as explant. This explant can be cells, can be tissues or pollen grain, or it can be a bark or a shoot tip, root tip, leaf part, right? All these can be taken as explant. Then there is a process of sterilization. These parts has to be sterilize. That part of the topic we will discuss in the procedure of plant tissue culture. Since it is an introductory topic, fine, I am giving the definitions, basic definition of this, and the terminology will be knowing in that. After explain, one more definition you try to understand. Here, one more word is known as callous. What do you mean by callous? The meaning of callous, the word meaning of callous is very slow, very lethargic, right? Lazy, right? You might have heard in somewhere, callous attitude, right? Slow attitude, right? Very lazy, very slow. Why is it named as callous? Because, fine, when explant is taken, fine, explant is taken on a nutrient media, for example, this is a bottle, you have a nutrient media here, fine, this explant is kept, fine. This is a very simple experiment. But you have to remember, you have to carefully done in all aseptic conditions by using many instruments, right? So when this explant is kept here, inoculated, in microbiology terminology we call it inoculation. Why? You are introducing it, inoculating it into the nutrient media. This nutrient media, in case of plant tissue culture, is mostly used as MS media. What media? MS media. Because these are the two scientists, one is known as Murashi and other is known as Skoog. The other is known as Skoog media. These two scientists designed the first nutrient media. They first formulated the nutrient media for plant for the growth of the plants. What exactly they did is, whatever the nutrients that are present in the soil, in the mud, right? Same nutrients they artificially added. Right? Temperature is maintained, pH is maintained. Clear? Macronutrients are added, micronutrients are added, like carbohydrates, proteins, vitamins. After that, minerals are added like calcium, magnesium, zinc, sulfur in very small quantities. After that, this plant growth promoters you have studied in your first year. Growth hormones, auxins are added. Fine. Cytokinins are added, gibberellins are added. Cytokinins are for the production of fruit, for the development of fruit, whereas axins and gibberellins are for the shoot induction. For these hormones are added in micro quantities. Vitamins are added, proteins are added, carbohydrates are added, fat are added. After that, such a complex nutrient media you prepare. That media you take in bottles. In that bottle, the explant, after taking the explant and after sterilizing this explant, right, and in 0.1% magnesium chloride, then it is washed in running water. After all that washing process is over, sterilization process of explant is over, in a aseptic condition, there is an instrument known as hot air oven where you sit, aseptic condition. Right? In that aseptic condition, this explant is transferred to that transfer process known as inoculation. After inoculation, this explant will be there in the plant, in this nutrient media in the bottle. These bottles under aseptic conditions of different temperature and light are maintained for a few days, for about 15 to 20 days. After these many days, from this explant, slowly the tissue will develop. Slowly the mass of tissue will develop, mass of cells like tumor will develop. Here, if I call this tumor, you don't get confused with the tumor of the cancer, what we have discussed in the 8th chapter. There also is a mass of cells. Here also, this is a mass of cells. What is callous? It's a mass of undifferentiated cells. Why we are calling them as undifferentiated? Because these cells are not shoot cells. These cells are not root cells. They are not leaf cells. They are not xylem and phloem tissue. They are not parenchyma or what. So they are not specified. They are not specified. They are not differentiated. Hence, they are generalized cells. What cells? Mass of generalized cells. Hence, we call this as the mass of. Now, I am coming to the definition of callous. What is callous? The mass of undifferentiated cells obtained from explant in plant tissue culture is what is known as callous. In the very beginning, I told you the meaning of callous. Very slow. Why? Here it takes around 15 to 20 days to grow, to differentiate into cells, mass of cells. Because it takes a very long period of time. Hence it is named as callous. Now, what is callous? The mass of undifferentiated cells obtained from explant in plant tissue culture is what is known as callous. So what is this? Right? Callous. So it is undifferentiated. After this, it has to be transferred to many other media, which has a shoot induction media or root induction media. Fine. All those things. Right. Now in today's topic, we have discussed about media, Murashi and Skoog. I will revise now. Fine. Now in today's topic, what we have discussed so far, we will revise now, already 15 minutes over. So in today's topic, we have discussed the definition of plant tissue culture, the process of growing of plants by using artificial nutrient media under aseptic conditions, under controlled conditions of light and temperature is what is known as plant tissue culture. This plant tissue culture was totally based on one property of plants, that is known as totipotency. Totipotency is nothing but the ability of any plant part to grow into a or to give rise to a complete plant of its own kind is called totipotency. The term totipotency was first coined by T.H. Morgan in the year 1901. It was practically proved by Gottlieb Heberlein in the year 1902. Hence, Gottlieb Heberlein is considered as the father of plant tissue culture. After that, I told you the process of plant tissue culture, then explant. What is explant? The previous part of the plant that gives rise to a complete plant. The previous part of the plant like cell, tissue or any part, any organ of the plant taken and processed in plant tissue culture is called explant. After that we come to callus. What is callus? The mass of undifferentiated cells obtained from explant in plant tissue culture is called callus. Now going back to this media part, nutrient media. So these two scientists, one is known as Morashig and the other is known as Skoog. They formulated the first media for plant tissue culture. So this was the most successful media, and this media is also known as basal media. Why? Because the very first step of plant tissue culture, this is the first media to be used everywhere, whether it's for shoot induction or root induction, or for to make transgenic plants, or for micropropagation. Okay, we'll talk about micropropagation also. This definition also will complete today. So these two scientists, one is known as Morashig, other is known as Skoog. So shortly referred as MS media. This MS media has all the constituents, whatever are there in the soil, in the sand, in the mud, in naturally whatever the nutrients are available. Same nutrients are artificially prepared and artificially added into a media having water, starch, carbohydrates, proteins, vitamins, right? After that minerals, right? Like magnesium, calcium, zinc, sulfur, phosphorus, and after all these, find different vitamins, vitamin A, vitamin B3, find folic acid, find NAA, all those, find gibberellic acid. So gibberellins, cytokinins, axins are added. So media are prepared. So after this media is prepared, explant is inoculated. After a few days, callus will grow. From this callus again, the callus will be taken. Then again shifted to different plants for different root induction, then shoot induction and all those things. After about 2 to 3 months in a bottle, you see a complete plant, and these plants are transferred to small racks and they will be marketed. Before that, there is a process we'll discuss. Now one more difference we will try to cover here, try to understand. One is known as micro-propagation. This will be many times it has asked in one mark in the part A question paper. Micro-propagation. Remember, the process of production of, last year also it has been asked in the biology for 1 mark, try to remember. The process of growing of millions of plants by using plant tissue culture which are genetically similar. Such a process is what is known as micro-propagation. And the plants which are involved here are known as somoclones. What do you mean by this? Their somatic is same. From the somatic fusion, somatic hybridization, one cell, fine protoplast is taken, the other protoplast is taken, they are fused, they are hybridized, and the plants you obtain from them are genetically similar. Such plants are known as somoclones. The plants are obtained from somatic fusion. I hope it's clear. Micropropagation also, the process of growing of millions of genetically similar plants by using plant tissue culture is what is known as micropropagation. In today's topic, we have discussed the general aspects of plant tissue culture, the historical part, the terminology involved, and the definition part. In the next part, we will discuss the process and the advantages of plant tissue culture. Keep studying. Do take very good care of yourself. God bless you. Good luck, take care. Do watch my class repeatedly. I will be uploading shortly on Tardigrade and YouTube also. If you have any doubts, contact me on my personal WhatsApp. Fine. And refer the study material. Keep studying. God bless you. Good luck, take care.